Hello, and welcome to Part 5 of Arcadia University's BI327 mini lecture series on nervous tissues. And in Part 5, we're going to focus in on physiology within the nervous system. Now, when we think about this, what we've got in the nervous system, our cells are going to be capable of neuronal signaling, capable of sending signals relatively rapidly throughout the body to control what the body is going to be essentially perceiving, as well as controlling what the body is going to be doing. And so if we take a look at neuronal signaling, what we're going to see is that there are going to be two primary ways in which that the nervous system is going to be sending signals within the body. The first is with an action potential. So in essence, we're looking at an electrical signal, and instead of looking at the flow of electrons, what we're going to be looking at is going to be the flow of ions. And so the action potential is going to be generated at the cell body, and it's going to be transmitted along the axon or along the length of a membrane. It's also possible that an action potential, call it slightly different, will be generated in the dendrites and carry that information to the cell body. But basically, we're looking at an electrical signal, looking at ion flow that's going to be able to carry a signal along the length of the membrane. The other type of neuronal signaling that's possible within the nervous system is going to be the use of neurotransmitters to essentially have a chemical signal so that you can essentially send a signal from one cell to the next cell. In at least adult mammalian tissues, adult human tissues, we don't have a lot of electrical synapses. So we don't have the ability for an action potential to go continuously from one cell to the next cell. And so what that means is we are dependent upon the ability of one cell to signal the next cell with a gap between them. And so we've got to have the ability to convert our electrical signal into a chemical signal, which then can diffuse across the space, activate that next cell, that target cell, and then convert that signal back into an electrical signal. And so that's the basis of neuronal functioning, the neurophysiology. And so if we take a look at this, well, the first thing we need to recognize is if we're going to look at these electrical signals, look at the ion flow, basically we need to have the ability to both have a difference in ion concentrations across the membrane and then be able to use that difference or kind of harness that difference in some way. Now, the important thing to recognize is that all living cells have a different ion concentration across their membrane. They're going to have a higher concentration of, say, sodium and chloride outside of the cell and a higher concentration of potassium inside of the cell. And that's going to be pretty much all cells within the body. What makes nerve cells different is that we're going to harness that difference, harness that concentration difference in a way that's going to allow us to send electrical signals across the cell. And so basically what's going to happen is when we generate this action potential, we're going to be looking at what's referred to as a voltage differential that passes along the length of the axon. The voltage differential basically means we're going to change the charge difference, change the ion concentration difference across the membrane, and that's going to spread down along the length of the axon. And so basically what's going to happen is we're going to open up ion channels, and these are going to be gated ion channels, so they're channels that are normally closed. We're going to cause them to open up. And so if you open up an ion channel, it's going to allow the flow of ions across that membrane. And so we're basically changing the permeability of the membrane. And we're not going to change it permanently. We're basically going to open up the channel for a short period of time, and then it's going to close right down. But when it's open, we're going to have the flow of ions down their concentration gradient, so that what happens then is instead of the normal distribution of charges, we're going to have a difference. We're going to have essentially a positive charge that's going to be generated. Okay. When that happens, that's going to trigger neighboring gated ion channels to open themselves, and it's going to cause the ion flow to spread, and it's going to cause that charge differential to spread. And so it's going to move down along the length of the membrane. What's going to happen behind that is that we're going to have pumps that are going to go through and restore things back to normal. And so in essence, what's going to happen is we're going to trigger an action potential. That action potential is going to open up some ion channels, and as it moves down the membrane, it's going to cause more and more ion channels to open to keep the action potential, to keep the flow of ions going as it moves down the membrane. Behind that, we're going to have pumps reestablishing that normal resting potential, reestablishing that normal concentration of ions across the membrane. Okay, so if we take a look at this, the flow of an action potential is essentially the flow of electrical signals, or at least the flow of ions, along the length of the axon, along the membrane. Now, in an unmyelinated axon, that impulse is a continuous wave. And so you open up some ion channels, and then you open up some ion channels in the next region of the membrane, and you open up some ion channels in the next region, and then some in the next region, and then some in the next region. And it's going to be continuous opening up every step of the way. It's a relatively slow process. Now, with a myelinated axon, instead of this continuous wave, we're going to have what's referred to as saltatory conduction. Basically means that the charge jumps from one node of Ranvier to the next node of Ranvier. So it's basically going to jump over that myelin covering, jump over that myelin sheath from one point to the next point. And by jumping from one point to the next point, it's going to be a more rapid signal. So basically what's going to happen is the myelin is going to be insulating the axon, blocking the flow of this ions, and we're going to have a very high concentration of ion channels at these nodes of Ranvier. And so we're going to build up a huge charge differential at one node of Ranvier, and it's going to be enough that as it spreads, it doesn't have to open up ion channels until it gets to the next gap, until the next node of Ranvier, where it's going to trigger a massive opening of ion channels and a very rapid depolarization of the membrane, flow of ions across the membrane, the charge differential, and it's going to be enough to trigger the next one. So it's basically going to jump from one point to the next point to the next point to the next point, and very rapidly send the signal within the nervous system. So unmyelinated and myelinated, same basic idea, opening up these gated ion channels and allowing the flow of ions down their concentration gradient, change the membrane properties, and allow for these electrical signals to spread. But in myelinated axons, it's going to move much more rapidly throughout the nervous system. Ultimately, it's going to get down to the tip of the axon. And at the tip of the axon, we're essentially going to have a space between the sending cell and the receiving cell. And so we've got to convert this electrical signal into a chemical signal. And so what we're going to have is on the presynaptic membrane, before the synapse, so we're looking at the sending cell, what we're going to have is an enlargement of the axon. It's referred to as the terminal bouton. And within that, what we're going to have is a collection of neurosecretory vesicles, essentially membrane-bound vesicles, which are going to be packed with the neurotransmitter. And so when that action potential comes down the axon, it's going to trigger the release of the neurotransmitter. So it's going to cause these neurosecretory vesicles, membrane-bound vesicles, to essentially fuse with the membrane through the process of exocytosis, dump out their neurotransmitter and release their neurotransmitter into that space. So that space is going to be referred to as either the synaptic cleft or synaptic gap or some variation of that. But it's basically about a 20 nanometer space between the sending cell and the receiving cell. It's going to be a fluid-filled space, which is going to allow the diffusion of chemical molecules, in this case chemical neurotransmitters, across the space. So the sending cell is going to dump out the neurotransmitter. It's going to diffuse across to the receiving cell. Now, there are going to be a lot of things going on within the synapse. The neurotransmitter can diffuse away. The neurotransmitter can be reabsorbed by the sending cell, or there's often going to be neurotransmitter enzymes. Enzymes are going to break down the neurotransmitters within the synaptic space because we want to be able to clear that up so that we can send another signal very, very rapidly. And so that's going to be a mechanism in place so that you send a signal, wipe the slate clean, and be ready to send another signal very quickly. On the postsynaptic membrane, post for after, so after the gap, after the synapse, looking at the receiving cell, we're going to have a thickening of the plasma membrane, thickening of the target cell. In many cases, it's going to wrap kind of partially around the sending cell's tip of the axon, the terminal bouton. And within this region, we're going to have receptors for the neurotransmitters, so they can bind to the neurotransmitter. Usually they're going to be coupled to an ion channel. So that what happens is the neurotransmitter binds to the receptor, cause the ion channel to open up, and the ion channels open up similar to what they did with the action potential. They open up, change the permeability of the membrane to the ion, and basically allow ions to flow down their concentration gradient. As the ions flow down their concentration gradient, they're going to cause that charge differential, or they're essentially going to establish or trigger that electrical signal. And that electrical change, that electrical signal, is going to cause the opening of nearby gated ion channels and cause this then electrical signal, this ion flow, to spread across the membrane. Now, if we take a look at how this can be affected in areas like muscle paralysis, we can go through and artificially affect the transmission of the signals. And so with something like botulinum toxin, you go through and you prevent the release of the acetylcholine. So you prevent the cells from releasing the neurotransmitter. A chemical like curare, a poison like curare, prevents the binding of acetylcholine to the receptors. So it basically blocks the receptors. You can send all the messages you want, but the receptors aren't able to bind to the neurotransmitter, and because of that, they can't open up their ion channels. In an autoimmune disease like myasthenia gravis, we essentially have the immune system of the body attacking the acetylcholine receptors. So it essentially goes through and destroys the receptors, destroys the muscle cells, so that we don't have the receptors being present that can receive these signals. So again, 3 different ways in which we can disrupt this signal and affect what's going on with the transmission from the nerve cell to the target cell, in this case the target cell being a muscle cell. Now that finishes up our lecture series on the nervous tissues. If you have any questions, feel free to email me at hoffmanj@arcadia.edu, and hopefully you'll be back for the next series of lectures. We're going to be taking a look at the muscular tissues.